Imagine, for just a moment, that you are suffering from a really severe stomach ulcer. The pain is just agonizing. It's this constant burning sensation right in your gut.
Oh, yeah, it's incredibly painful.
Right. And let's say it's the 1970s or maybe the early 80s, and you finally go to your doctor hoping for a cure. You know what they tell you. They look you in the eye and essentially say that it's your own fault.
Yeah, they blame the patient entirely.
Exactly. They'd say you have a highly stressful job or, you have a type A personality, you eat way too much spicy food, drink too much coffee, and all that anxiety is causing your body to produce an excess of stomach acid.
Acid that they thought was literally burning a hole right through your stomach lining.
Right. So for decades, the standard medical advice was just to calm down, go on this bland diet of chalky milk, and take expensive acid-suppressing medication for the rest of your life.
Which is just wild to think about now.
It is. Because years later, you find out that the entire medical establishment was completely, undeniably wrong. Your ulcer wasn't caused by your lifestyle or your stress or your diet at all. It was actually caused by a highly specialized, stealthy bacterium.
It honestly remains one of the most stunning medical reversals of the 20th century. I mean, we are talking about a complete paradigm shift that fundamentally altered how we understand the human body, the role of microbes, and really the very definition of a disease.
And that is exactly our mission for today's deep dive. We are exploring this microscopic survivalist known as Helicobacter pylori, or just H. pylori.
It's a fascinating organism.
It really is. We've gathered a huge stack of research notes for today covering the microbiology, the completely wild history of its discovery, and the really intense ongoing debate surrounding this bug.
Because it's not a settled science, even today.
Not at all. So, we are going to figure out how this bug manages to survive the body's ultimate acid trap, how it forces us to rethink modern medicine, and importantly, why all of this matters directly to you and your own health. Okay, let's unpack this. To really appreciate how groundbreaking this bacterium is, we first have to understand the absolute, impenetrable scientific wall of dogma that it crashed into.
You really have to put yourself in the mindset of a gastroenterologist back then. The stomach was viewed as this highly hostile, completely sterile environment.
It's just totally empty of life.
Exactly. The lumen, which is the hollow space inside your stomach where food actually lands, has a resting pH level that normally sits somewhere between 1.5 and 3.5.
Which is basically battery acid. I mean, that is an environment biologically designed to break down a tough piece of steak, right?
Oh, absolutely. It's meant to kill pathogens and protect the rest of your digestive tract. So the idea of a tiny, fragile microorganism setting up a permanent home in there, well, it seemed biologically impossible.
Yeah, I imagine they'd think you were crazy for suggesting it.
It was considered absurd. Stomach acid is potent enough to dissolve most bacteria within minutes. So medical science firmly, and I mean dogmatically, believed that no resident bacterial life could possibly exist there.
But didn't pathologists ever see anything under the microscope?
Well, they did. But if a pathologist happened to look through a microscope and actually saw bacteria on a stomach tissue sample, they dismissed it immediately as a contaminant.
Oh, wow, so they just ignored it.
Yeah, they assumed it was just an artifact from the lab preparation, maybe something introduced by a dirty instrument, but definitely not something actually living and multiplying inside a living patient.
Right, because under that belief system, doctors had to find some alternate explanation for ulcers. If it wasn't a bug, it just had to be a lifestyle disorder, a physiological defect.
And the treatment was just to manage those symptoms forever.
Exactly. You had these massive blockbuster drugs like Tagamet and Zantac, which suppressed acid production. And those weren't just common medications, right? They were among the best-selling, most lucrative drugs in the entire world.
Oh, billions of dollars.
Yeah. The entire pharmaceutical industry is basically built on the premise that ulcers could only be managed, never actually cured.
What's fascinating here is the sheer scale of the institutional blindness. When two researchers in Australia, a pathologist named Dr. J. Robin Warren and a young internal medicine resident named Dr. Barry Marshall, when they started challenging this in the early 1980s, they were met with extreme hostility.
I can imagine.
Yeah.
Taking on $1 billion dogma.
Right. In 1979, Dr. Warren started noticing these thin, curved bacteria in stomach biopsy samples from patients who had gastritis.
Which is a really severe inflammation of the stomach lining, right?
So correct. But he couldn't get anyone to take him seriously at all. So in 1981, he teamed up with Dr. Marshall to try and prove these bacteria were real.
But just seeing a shape on a glass slide isn't enough to convince the medical establishment. To prove a bacteria is a living infectious agent, you actually have to be able to grow it in a lab, don't you? You had to culture it. And from the notes we have here, it looks like they failed at doing that over and over again.
They hit a total wall for months. The standard protocol in microbiology labs at the time was to leave culture plates in the incubator for 2 days.
Just 48 hours.
Yeah, and if nothing grew after 48 hours, the technician just assumed the sample was sterile and threw it in the trash. Normal bacteria, like E. coli or staph, they will form massive, visible colonies in a matter of hours.
Oh, I see.
But every single biopsy sample Warren and Marshall tried to grow came back completely empty.
So what changed? Did they figure out, like, a new chemical broth to feed it or something?
It was actually an incredibly fortunate lab accident.
Really?
An accident. Yeah, it was the Easter holiday in 1982. A batch of these biopsy plates was sitting in the incubator, and the lab technician who normally cleared them out was actually away for the long weekend.
Oh, so they just sat there.
Exactly. The plates were accidentally left incubating for 5 full days. When they finally checked the plates on Tuesday, the organism had absolutely thrived. They had these visible, translucent colonies of a previously unculturable bacterium. It turned out H. pylori wasn't impossible to grow. It just grows incredibly slowly compared to other bugs.
That is amazing. An Easter miracle in a petri dish.
Yeah.
But even with the physical bacteria growing in the lab, publishing their findings in 1983 and 1984 didn't suddenly change everyone's minds, did it?
Not at all.
Because you can't really blame the establishment for their skepticism, right?
Mm-hmm.
It's like someone telling you they found a thriving colony of fish living inside an active volcano. The stomach is designed to melt things. The medical community must have looked at Warren and Marshall like they had completely lost their minds.
Oh, they were essentially laughed out of conferences. The medical establishment's resistance was protected by this deeply entrenched paradigm, this no acid, no ulcer doctrine.
Right, because of all the money in textbooks.
Exactly. You had entire careers and a massive pharmaceutical market invested in the idea that stress and acid were the sole culprits. To upend that, Warren and Marshall needed human trials to prove causation.
But I'm guessing they couldn't get approval for that.
Right, because their theory was considered fringe science. They couldn't get the funding or the ethical approval to test it on humans.
Which leads us to 1984. They are sitting there with this groundbreaking discovery, totally stonewalled. And Barry Marshall decides to take matters into his own hands. And the way he did it, he literally drank a beaker of bacteria. That is wild. It sounds less like the scientific method and more like a dare.
It really does.
Yeah.
But yeah, he performed what is now considered one of the most famous and dangerous self-experiments in medical history. Marshall took a beaker filled with a murky broth culture teeming with billions of living H. pylori cells, and he just drank it.
I still can't get over that. He intentionally infected himself with an unknown pathogen just to prove a point. Did he get an ulcer immediately?
Well, he actually expected it would take years for an ulcer to develop, if one developed at all. But the biological reaction was violent and immediate.
No, like within days.
Yeah. Within a few days of drinking the broth, Marshall developed profound nausea, exhaustion, and he began vomiting. He had triggered acute gastritis.
His body was fighting back.
His immune system was suddenly at total war. He then underwent an endoscopy and a biopsy, which definitively confirmed 2 things. First, his stomach, which was previously perfectly healthy, was now swarming with H. pylori. And second, the tissue was severely inflamed.
He actually proved it.
He did. And after proving the point, he just cured himself with a simple course of antibiotics.
That is an incredible level of dedication. He made himself the literal guinea pig.
But beyond the dramatic flair of it all, what did that actually prove to the broader scientific community?
It rigorously fulfilled classical disease causation criteria. There's a framework known as Koch's postulates.
Right, the gold standard for infectious disease.
Exactly. To prove a microbe causes a disease, you have to isolate it from a sick patient, grow it in a lab, introduce it into a healthy host to cause the exact same disease, and then re-isolate it from that newly sick host. Marshall's self-experiment hit every single requirement. It was airtight.
Totally airtight. He provided undeniable proof that a specific microorganism could single-handedly take a healthy stomach and turn it into a sick one. This act, along with mounting evidence over the next decade, eventually broke the dam.
So the establishment finally gave in.
Yeah. In 1994, the U.S. National Institutes of Health issued a consensus statement recognizing H. pylori as a major cause of peptic ulcer disease. And then, in 2005, Marshall and Warren won the Nobel Prize in Medicine.
So they went from being the crazy guys in the corner to Nobel laureates.
Oh.
But, this immediately brings up a massive biological contradiction. We just talked about how the stomach is a vat of battery acid,
Yeah.
and we know H. pylori survives in it. How on earth does a fragile, single-celled organism pull off this impossible engineering feat?
It is brilliant. The mechanism is a two-part defensive strategy. The first part is a biochemical trick involving an enzyme called urease.
Urease, okay.
Yeah. H. pylori dedicates an enormous amount of its biological energy to producing urease. It actually makes up several percent of the bacterium's total protein weight.
That's a huge investment for a single cell. What exactly is that enzyme doing to keep the acid away?
It basically acts as a localized acid neutralizer. There is a chemical compound called urea naturally present in your stomach fluids. When H. pylori's urease enzyme comes into contact with that urea, it breaks it down into carbon dioxide and ammonia.
Wait, ammonia? But ammonia is highly basic, right?
Exactly. Ammonia is highly alkaline. So by constantly pumping out this ammonia, the bacterium creates a personal walkable pH bubble around itself.
Oh, wow.
The surrounding stomach fluid might be a lethal acid bath, but inside that microscopic bubble, the environment is perfectly neutralized and safe.
That is wild. It essentially generates its own chemical force field. And looking at the notes, this specific survival mechanism is actually how modern medicine tests you for the infection today, isn't it?
It is, yeah. It's called the urea breath test, and it is an elegant application of this exact biology.
How does that work?
Well, if you go to the clinic with stomach pain, the doctor might have you drink a specialized solution of urea that has been tagged with a harmless carbon isotope.
Okay, so you drink the tagged urea.
Right. If you have H. pylori in your stomach, its urease will immediately break down that tagged urea. The tagged carbon dioxide then enters your bloodstream, travels to your lungs, and within 15 to 30 minutes, you will literally exhale it.
And the doctor just measures your breath?
Precisely. If there is no H. pylori, there is no urease, the urea stays intact, and you don't exhale the tagged CO2.
I love when the science translates so perfectly to the clinic. Okay, so it has this chemical force field, but it can't just float around in the middle of the stomach acid forever, right? It needs to find a permanent home.
Yeah, the force field is only designed to keep it alive long enough to reach a sanctuary. H. pylori is what microbiologists call a microaerophile, meaning it requires an environment with very low levels of oxygen.
So the open stomach is too exposed.
Way too exposed. It doesn't want to fight the acid in the open hollow of the stomach. Instead, it heads straight for the thick layer of mucus gel that coats the inner walls of your stomach.
Just to clarify for the listener, this mucus layer is the biological barrier that protects our own stomach cells from being digested by our own acid. It's this thick, heavy jelly. How does a tiny bacterium swim through something that thick?
It relies entirely on its physical shape and motility. H. pylori isn't round or rod-shaped like a lot of common bacteria. It actually has a distinctive corkscrew shape.
Literally like a drill.
Exactly. And it is also equipped with 4 to 6 sheathed flagella, which operate like tiny, whip-like outboard motors on the end of the cell.
Oh, wow.
It uses these tails to physically screw its way right through that heavy, viscous mucus. It drills away from the acid lumen and migrates toward the neutral, highly hospitable zone right next to your stomach wall.
That's incredibly specialized.
It is, and we know this mechanical movement is essential because mutant strains of H. pylori that are bred without these flagella are completely unable to establish an infection. They just get trapped in the mucus and die.
So it's basically an astronaut wearing an anti-acid spacesuit, drill biting its way through a wall of protective jelly.
That is a highly accurate way to visualize it. Yeah, it drills through the danger and sets up camp right on the surface of your stomach cells.
Surviving the stomach is a great defense. But we really need to understand how this squatter actually damages the host. How do we go from an organism just trying to survive to a disease mechanism that causes excruciating ulcers?
Well, once it gets through the mucus, it anchors itself to your cells. It uses specialized adhesion proteins. The most studied one is called BABA, which basically act like molecular grappling hooks.
Grappling hooks? That sounds aggressive.
Yeah, they bind directly to specific sugar molecules on the surface of your stomach cells, specifically the Lewis B antigen. Once it is physically locked on, the defense phase ends and the attack really begins.
What does it do?
It deploys a structure called a type 5e secretion system, which operates exactly like a microscopic syringe, injecting bacterial toxins directly into your healthy cells.
A molecular syringe. What kind of payload is it injecting?
There are 2 primary toxins that do the heavy lifting here. The first is called CAGA.
Okay.
When CAGA is injected into a stomach cell, it hijacks the cell's internal communication. It disrupts how the cells bind to each other and forces the host cells to multiply abnormally. And this cellular chaos is heavily associated with an increased risk of gastric cancer.
That's terrifying. What's the second toxin?
The second is Vitae. This one doesn't even need the syringe. The bacterium just secretes it into the surrounding area. Vitae binds to the host cells and forms actual pores-like physical holes in the cell membranes.
It punches holes in our cells.
Yeah. It creates large, empty bubbles called vacuoles inside the cells that eventually crush the cell from the inside out. Furthermore, vacay actively suppresses your local immune system.
Which explains how it survives so long.
Exactly. That immune suppression is how this infection can persist in your stomach for decades without your body clearing it out.
Here's where it gets really interesting, because when you say ulcer, the listener is probably picturing this bacterium taking actual bites out of the stomach lining, physically chewing a hole in the tissue. But looking at the sources, that's a massive misconception. So the bacteria isn't chewing through the stomach lining, it's poking the bear. And our own immune system's chronic inflammation is what burns the house down.
That is a brilliant analogy, yes. H. pylori does not consume your stomach tissue. The ulcer is a side effect of your body's own chronic low-grade inflammation.
So it's basically friendly fire.
Completely. Your immune system detects these toxins, recognizes an invader, and continuously sends in inflammatory cells to fight it. Over years and decades, this sustained inflammation degrades the protective mucus layer and the epithelial defenses, the actual layer of cells lining your stomach.
And the type of damage depends entirely on where the bacteria sets up camp, doesn't it?
Correct. If the infection is concentrated in the lower part of the stomach, an area called the antrum, the localized inflammation actually interferes with the hormones that regulate acid.
So it messes with the thermostat, basically.
Right. It causes your body to spike acid production, dumping excessive amounts of acid down into the duodenum, which is the first part of the small intestine. That excess acid erodes the tissue, causing a duodenal ulcer.
And what if it's not just in the lower part?
Conversely, if the infection spreads throughout the entire stomach, a condition called pangastritis, it actually destroys the acid-producing cells. This lowers your acid levels, but severely accelerates cellular mutation. That widespread inflammatory pattern is what leads to gastric ulcers and, more terrifyingly, gastric cancer.
Which is a very real threat. I mean, back in 1994, the World Health Organization officially classified H. pylori as a Group 1 carcinogen. That places this bacterium in the exact same formal risk category as asbestos, plutonium, and tobacco smoke.
If we connect this to the bigger picture, that carcinogenic classification is exactly why the discovery by Warren and Marshall is considered such a triumph of modern medicine.
It saves so many lives.
Countless lives. Because of them, if you have an ulcer caused by H. pylori today, you aren't told to drink milk and manage your stress while the cancer risk quietly grows. You are cured.
Cured completely, not just managed like they used to do.
A definitive cure. Gastroenterologists use a 1 to 2 week course of targeted antibiotics, usually a triple or quadruple therapy that combines different antibiotics with an acid suppressor.
To hit it from all sides.
Exactly. The acid suppressor calms the environment, the antibiotics eradicate the bacteria, the chronic inflammation finally stops, and the ulcer heals permanently.
That is incredible. But, this leads us to the final and maybe most confusing part of the deep dive. We've established that it's a Group 1 carcinogen. It uses a molecular syringe to inject toxins. It causes ulcers, and it leads to cancer. But there is a massive biological paradox here in the notes.
There really is.
If this bug is so deadly, why do the vast majority of people who are infected with it never actually get sick?
That remains one of the most compelling mysteries in modern microbiology. First, we need to completely discard the idea that this is some new plague. H. pylori is an ancient organism.
Like how ancient?
It has co-evolved with humans for tens of thousands of years. We know this because scientists actually found H. pylori DNA inside the stomach of Ötzi, the Copper Age mummy discovered frozen in the Alps who lived over 5,300 years ago.
Oh, wow. So it's been a passenger in the human gut since the very beginning.
Yes. This bacterium migrated out of Africa alongside early human populations, and because of that long co-evolution, 80 to 90% of people infected with H. pylori are completely asymptomatic.
80 to 90%.
Yep. They carry this Group 1 carcinogen for their entire lives and never develop an ulcer and never get cancer. Whether you get sick depends on a really complex interplay of your own genetics, environmental factors like your diet and whether you smoke, and the specific strain of the bacteria, for instance, whether it is armed with that Kaga toxin we discussed earlier.
Okay, so for most people, it's harmless. But there is a fierce debate happening in the scientific community right now, spearheaded by a microbiologist named Martin Blaser. It's called the Blaser paradox, and it goes way beyond the bacteria just being harmless.
It does. Blaser and other researchers have observed a fascinating epidemiological trend. Over the last century, as wealthy nations have improved sanitation, reduced household crowding, and heavily utilized broad-spectrum antibiotics, the prevalence of H. pylori in the population has plummeted.
Which sounds like a huge win.
It is. Consequently, and thankfully, rates of gastric cancer have fallen dramatically. However, at the exact same time, rates of gastroesophageal reflux disease, or GERD, and esophageal adenocarcinoma, which is a deadly type of throat cancer, have risen sharply.
Wait a minute. Are you telling me this group 1 carcinogen, this ultimate villain that we've been praising researchers for discovering and killing, might actually be a part-time superhero? How could it possibly prevent throat cancer?
Blaser's hypothesis provides a mechanical explanation for it. Remember how we discussed that widespread H. pylori infection can actually lower stomach acid production by damaging those acid-producing cells?
Yeah, the gastritis.
Well, if you completely eradicate H. pylori from the human microbiome, the stomach might produce excess acid unchecked. That unregulated acid frequently splashes up into the esophagus, causing severe GERD and, over time, triggering cellular mutations in the throat that lead to esophageal cancer.
That's a really dark trade-off.
Blaser argues that because we co-evolved with this organism, it acts as a crucial regulator of our immune system and our acid levels, conferring actual protective benefits.
I mean, I had to push back on that a little bit. Correlation is not causation, right? Just because GERD goes up as H. pylori goes down doesn't necessarily mean the bacteria was actively preventing the GERD. Couldn't the rise in reflux and throat cancer just be due to modern diets, obesity, and processed foods?
That is the exact counter-argument many oncologists make, which is why this is such a heated, active debate right now. But Blaser's camp argues it is more than just a statistical correlation. They are finding actual mechanical links showing how H. pylori modulates immunological responses, potentially protecting against not just esophageal diseases, but also certain childhood asthmas and allergic conditions.
So it ties into the whole hygiene hypothesis.
Exactly. It fits into the broader hygiene hypothesis, which suggests that by exterminating the ancient microbes we evolved alongside, our immune systems become hypersensitive and unregulated.
This active clinical tension must make public health decisions incredibly difficult. I mean, if you were a doctor, do you wipe it out to prevent stomach cancer or leave it alone to prevent throat cancer?
It heavily dictates major public health strategies today. In regions like East Asia, where highly virulent strains of H. pylori make gastric cancer a leading cause of death, the strategy is aggressive. They launch mass campaigns to test and treat everyone.
Which makes sense given the risk.
Right. But in Western countries, knowing that mass antibiotic use disrupts the broader gut microbiome and might increase esophageal risks, doctors are shifting toward targeted, risk-stratified approaches. You test the strain, you look at the patient's family history, you evaluate their symptoms, and you make a nuanced individual decision on whether the bug actually needs to die.
So what does this all mean? For decades, millions of people suffered in agonizing pain under the illusion that their own bodies were failing them because they couldn't handle the stress of daily life. The story of this stealthy bacterium forces us to practice profound medical humility. What we definitively knew about stress, diet, and ulcers was completely, entirely wrong. But the good news is, for you, if you ever have a persistent, unexplained stomach issue, science now has the exact tools, like that brilliant urea breath test, to find and permanently cure the underlying biological infection.
This raises an important question. How many other chronic diseases, conditions we currently blame on modern lifestyle, aging, or genetics are actually the result of ancient, stealthy microorganisms that we simply haven't learned how to see yet? H. pylori changed the paradigm of gastroenterology forever, proving that sometimes the answer is hiding right in front of us, wrapped in an anti-acid spacesuit.
It really makes you look at your own body completely differently. You aren't just an individual. You are a complex, walking ecosystem. And as we leave you today, consider this final thought. As modern medicine gets better and better at using powerful antibiotics to wipe out ancient microbial hitchhikers like H. pylori, we have to ask ourselves a difficult question. Are we carelessly deleting lines of evolutionary code from the human microbiome without fully understanding the software? Next time you are prescribed an antibiotic, take a moment to think about the ancient, delicate ecosystem inside you that you are permanently altering. Thank you for joining us on this deep dive.